The main point of introducing mitosis is actually to contrast it with meiosis. Whereas mitosis starts with, at the very beginning, two copies of every chromosome in the cell, and then eventually leads to the creation of two daughter cells that also have two copies of every chromosome in every cell. Meiosis actually separates the different chromosomes, so you end up with just a single copy of every nucleus of every chromosome in every cell. And the way this happens is through a process called synapsis. So you undergo a lot of the same processes in meiosis. You begin with DNA replication. You undergo you dissolve the nucleus, and then having dissolved the nucleus, you end up, rather than aligning every chromosome pair along the every chromosome along the metaphase plate, you end up pairing the now replicated DNA along the metaphase plate. And so your two copies of chromosome 1 are going to line up together. And in the process of lining up, they undergo what's called synapsis. Synapsis is where you get genetic recombination. This is where you get genetic exchange. This is where your mother's chromosome and your daddy's chromosome come together and exchange genetic information. This is what you would refer to as molecular sex. It's where the actual exchange of genetic information occurs. Having undergone synapsis, you then have a single division where now you still have two copies of every chromosome in the daughter cells, but then you undergo a second division, and the result of the second division are 4 haploid daughter cells. So you start off with 2 copies of in the mother cells, and the result after a series of replication, synapsis, and a second division are 4 haploid daughter cells. These haploid daughter cells are normally the sex cells.